Welcome back to G Fun Facts Online, the channel where we peel back the layers of the world's most complex systems to understand how they really work. I am your host, and today we are diving into one of the most significant breakthroughs in the history of medicine. We are going to talk about a paradigm shift that is changing the way we think about life, death, and the war on cancer. For decades, the pillars of oncology were immutable. If you were diagnosed with cancer, your options essentially boiled down to three things: surgery to cut it out, radiation to burn it out, or chemotherapy to poison it out. While these modalities have saved countless lives, they share a common flaw. They are blunt instruments. They attack rapidly dividing cells indiscriminately, often causing profound collateral damage to healthy tissue, leading to the hair loss, nausea, and immune suppression we associate with cancer treatment. But the 21st century has witnessed the birth of a fourth pillar. This new approach does not rely on external poisons or radiation beams. Instead, it relies on the internal intelligence of the human immune system. We are talking about chimeric antigen receptor T-cell therapy, or CAR-T for short. CAR-T represents a total departure from pharmaceutical precedent. It is not a chemical compound metabolized by your liver. It is what scientists call a living drug. Once infused into a patient, these genetically engineered cells do not merely circulate and fade away like a standard medication. They hunt, they recognize, they kill, and crucially, they reproduce. A single infusion can expand into an army of billions within the patient's body, remaining on patrol for years, potentially offering a functional cure for cancers that were once considered terminal. Today, we are going to break down the intricate science, the harrowing development history, the complex manufacturing processes, and the future trajectory of this technology. To understand CAR, we first need to understand the T-cell. Think of the T-cell as the foot soldier of your adaptive immune system. These are hunter-killers equipped with sensors called T-cell receptors. Their job is to scan the surface of other cells for evidence of infection or mutation. However, cancer is insidious. Because cancer arises from the body's own tissues, it often learns to cloak itself from this surveillance. It can hide the molecular flags that T-cells use to identify threats. This is where the bioengineering comes in. The chimeric antigen receptor, or CAR, is the solution to this evasion. The term chimera comes from Greek mythology, referring to a beast that was a combination of a lion, a goat, and a snake. In biology, it means a synthetic molecule, combining parts from different sources to create a new function. A CAR construct is a masterpiece of protein engineering composed of three critical domains. First, you have the extracellular domain, or the sensor. This is usually a single-chain variable fragment derived from a monoclonal antibody. Unlike a natural T-cell receptor, which requires a threat to be presented on a silver platter by the cell, this sensor allows the CAR-T cell to latch directly onto a protein on the surface of a cancer cell, acting like a high-powered magnet. For most blood cancers, this target is a protein called CD19 or BCMA. Second, there is the transmembrane domain, which is the anchor. It structurally connects the outside sensor to the inside machinery of the cell. And third, and perhaps most importantly, is the intracellular domain, the engine. This is where the magic happens. When the sensor latches onto a cancer cell, this internal domain triggers the T-cell to activate. In the early days, first-generation CARs only had a basic signaling chain called CD3 zeta. They could activate, but they lacked staying power and died out quickly. The breakthrough came with second-generation CARs, which added a co-stimulatory domain, typically either CD28 or 4-to-1BB. This acts like a turbocharger, allowing the T-cells to proliferate rapidly and survive for long periods in the patient. When a CAR-T cell engages a tumor cell, it sends an electric jolt of activation signals. The T-cell then releases a payload of perforins and granzymes. Think of these as molecular grenades that punch holes in the cancer cell and induce apoptosis, or programmed cell death. Simultaneously, the T cell begins to divide, making copies of itself to join the fight. Now, how do we actually make this drug? This is where things get incredibly complex. Unlike a pill pressed in a factory, every dose of autologous CAR T therapy is a bespoke creation, manufactured from the patient's own blood. This is a process known as vein-to-vein manufacturing, and it is a logistical high-wire act that typically takes 2 to 4 weeks. It starts in the hospital with a procedure called leukapheresis. Blood is withdrawn from the patient and passed through a machine that separates out the white blood cells while returning the red blood cells and plasma to the body. These collected cells are then frozen and shipped to a central manufacturing facility, often owned by pharmaceutical giants like Novartis, Gilead, or Bristol-Myers Squibb. Once they arrive at the factory, the cells are thawed and activated. Scientists use artificial dendritic cells or magnetic beads to wake the T cells up. Then comes the genetic transduction. This is the critical engineering step. A viral vector, usually a deactivated lentivirus or gamma retrovirus, is used to infect the T cells. These viruses have been gutted of their reproductive machinery and serve only as delivery trucks. They insert the genetic code for the CAR construct directly into the T cell's DNA. Once integrated, the T cell permanently reads this code and begins building CAR receptors on its surface. But a few million cells aren't enough. We need an army. So step 4 is expansion. The engineered cells are placed in bioreactors and fed a rich soup of nutrients and growth factors like interleukin-2. Over several days, they multiply into hundreds of millions of potent CAR T cells. Finally, the product is washed, formulated, frozen, and shipped back to the treatment center. The patient undergoes a round of lymphodepleting chemotherapy to clear out their existing immune system to make space, and then the thawed CAR T cells are infused into their bloodstream. Since the historic approval of Kymriah in 2017, the clinical landscape has exploded. Kymriah, developed by Novartis and the University of Pennsylvania, uses the 4:1 BB co-stimulatory domain, which promotes a slower, longer-lasting expansion. It famously saved Emily Whitehead, the first pediatric patient, who remains cancer-free over a decade later. Then, there is Yescarta, developed by Kite Pharma. It uses the CD28 domain, which induces a more rapid, explosive attack on the tumor, making it a heavyweight treatment for aggressive lymphomas. We also have newer entrants like Breyanzi, which infuses a defined 1:1 ratio of helper and killer T cells for a more controlled response, and Carvykti, a highly potent therapy for multiple myeloma that targets the BCMA antigen with incredible efficacy. But we must be clear, this power comes with significant risks. When billions of T cells engage their targets simultaneously, they unleash a biological storm known as cytokine release syndrome, or CRS. As the T cells kill cancer, they release massive amounts of inflammatory cytokines like interleukin-6. This causes high fevers, low blood pressure, and in severe cases, organ failure. Interestingly, the antidote to this is a drug called tocilizumab. It was originally an arthritis medication that blocks the IL-6 receptor. Its discovery as a rescue drug for CRS was serendipitous, but it is now the standard of care, acting like a fire extinguisher for the immune system. There is also a risk of neurotoxicity, where patients become confused or lose the ability to speak due to inflammation breaching the blood-brain barrier. Furthermore, because the target antigen CD19 is found on healthy B cells as well as cancerous ones, CAR T therapy destroys both. This leaves patients with B-cell aplasia, meaning they cannot make antibodies and may require immune globulin infusions for life. The current frontier is the challenge of solid tumors. While blood cancers have melted away under CAR-T, solid tumors like lung, breast, and pancreatic cancer have remained stubborn. Solid tumors present three major barriers. First, the trafficking problem. T cells must exit the bloodstream and physically penetrate dense tissue. Second, antigen heterogeneity. Solid tumors are patchy. Some cells have the target, others don't. If you kill the positive ones, the negative ones just grow back. And third, the hostile microenvironment. Solid tumors build a fortress around themselves that is acidic, low in oxygen, and filled with immunosuppressive chemicals that shut down T cells. To combat this, scientists are developing armored cars that secrete their own shields to survive this environment, and cars that release enzymes to dissolve the tumor's physical matrix. Looking to the future, we are moving toward off-the-shelf or allogeneic CAR-T cells. These are created from healthy donor blood, using gene editing tools like CRISPR to remove the donor's natural receptors, preventing rejection. This would allow hospitals to keep frozen vials in stock for immediate use, drastically reducing costs. We are even looking at in vivo engineering, where we inject viral vectors or lipid nanoparticles directly into the patient to reprogram their T cells inside their body, skipping the lab entirely. The trajectory is clear. We have moved from poisoning cancer to outsmarting it. For thousands of patients who had run out of options, these engineered cells have given back a lifetime. As the technology matures, living drugs may well become the standard of care, turning the terrifying diagnosis of cancer into a manageable and curable condition. Thanks for listening to G Fun Facts Online. Stay curious.